So the big question, I guess, for my teaching lessons there, trying to think of something that would be relevant to people in this building is, how much fish do groups of marine mammals eat? It's a big question. It's a central question to a lot of things. The estimates the scientists throw out. So I'm going to present the current state of knowledge of how we come up with these estimates, and also the historical background. And the history is important for a couple of reasons, I feel. Partly because so we know why do we now do things this way, what was the progression that got us to this point, and that tells us about the strengths and weaknesses of what we're doing today. And also, I think history is neat. That's where all the tidbits are. And also, I think professionally it's important to know where you come from your chosen profession, what went on behind. This isn't all stuff that was created in the last 10 years. So when we're talking about estimating food requirements for a group of animals, in this case marine mammals, we're talking about the science of bioenergetics. So the first thing we need to define, what is bioenergetics? There's two possible ways to define it. One is the flow of energy through a biological system, whether that's an individual animal or an ecosystem. The second definition of bioenergetics is an alternative psychotherapy that's based on the connection between mind and body. I'm probably not going to get to that today. So, what are the goals of today's lessons? It's to understand the steps that are used or required to come up with an estimate of how much food a marine mammal or a population of marine mammals are using, to have an appreciation of the natural variation around those point estimates, to understand it's not a single estimate, no matter how precise or accurate it might be, there's a lot of natural variability in that system, and that's important to understand and take into account. And the other thing, perhaps more important, is to understand the strengths and weaknesses behind the methods that we use to come up with these food estimates, so that when you hear a number that is derived somehow, you have an idea of how accurate or how precise that number might be, or what are some of the additional questions you might have to ask for how did you come up with that number. So, how much fish, how much prey, but we're really talking about how much fish in most cases a marine mammal population consumes, is a big question in the popular media. Here's some quotes just from relatively recent newspaper articles talking about the impact that marine mammals have, how much fish they're taking out of the oceans. And it's not just important from a scientific system, you know, if we're doing ecosystem modeling within this building, but also statements like this catch the public's eye, and they're going to lead to policy decisions as far as resource management goes. Now, even before we had any hard data, you know, fishermen and scientists knew that we didn't know how much fish populations of marine mammals consume, but we knew it was a lot. You know, they've been characterized as voracious consumers of fish flesh. These biological vacuums consuming huge swarts of commercially important fish. And that's kind of the key. They were commercially important. So we saw them as this competitor that were taking way more than their share of fish in the ocean. There's various scientific justifications that we tried to base this on. You know, the idea that they had to thermoregulate in cold oceans, so they had to eat more, which would suggest they're maladaptive. But really, we saw them going directly after our fish, whether it was trawling or hook and line, or whether they were appearing in our fishing net, or whether we're boating along and we see a seal or a group of seals swarming some salmon stocks, or having this huge, great big salmon in their mouth at the surface and shaking in their heart. So they have this concept that marine mammals eat way more than the same sort of biomass of terrestrial mammals. And because of the competition, you know, we felt, well, we have to manage these marine mammals. And management of marine mammals generally means putting a bullet in their skull, because we want to limit the amount of our fish that they're taking out of the ocean. What's interesting is, even recently, when we're talking about managing the oceans for the benefit of marine mammals, so making sure there's enough Chinook salmon for the southern resident killer whale population, there are still people talking about killing other marine mammals in order to provide the fish for this group of marine mammals. And that's because they're having a huge impact. Now, when you're trying to manage an ecosystem, if you're at some meeting, you know, inevitably the question is going to arise, well, how much fish do they eat? This is a central question for managing the system, and they need an answer. And in the early days, we really had no idea. And you wouldn't think it would be that difficult an answer to come up with. I mean, it's a pretty simple formula on paper. You look at how much energy an individual marine mammal needs. You divide it by what you're going to get for every fish it consumes. So that tells you how many fish per individual. You multiply that by the population size, and wham bam, you got the food intake for that population. Pretty simple. Well, we're going to show you, I think, today that it's not so simple. We didn't have a lot of science starting off to answer things like what are the individual energy needs of a marine mammal. We just had these assumptions. And so that's where I'm going to focus a lot of my talk on. But we're also going to delve a little bit into what does a marine mammal actually get out of a fish, because that's an important consideration. The science really began probably around the mid-1980s at the University of Guelph. And they started measuring something called resting metabolic rate, or resting metabolism, using something called gas respirometry. And there's three important points here. One, University of Guelph, shout out because that happens to be my alma mater. Keith Ronald is the guy in the dark suit looking concerned about some of the seals that he brought in to the laboratory at Guelph to measure resting metabolic rate using this methodology. So I want to talk a little bit about it. Basically, gas respirometry is a way of measuring, estimating energy consumption by looking at oxygen consumption, which a lot of us take for granted now, but the history of it is kind of interesting, or I think it's interesting. And to start off with two Frenchmen in the late 1700s, Antoine Lavoisier and Pierre-Simon Laplace, I apologize for my horrible French accent, who, after stealing some ideas from some Scottish scientists, realized that the work done by living organisms is basically expressed as heat. And so they said, well, if we can quantify that heat, we know how much work we're doing. And so they built a calorimeter, so something for measuring calories. And it was essentially an insulated chamber surrounded by ice. And because you knew how much energy it took to melt a certain amount of ice, you could put an animal in there. In their case, it was a guinea pig. Put it in for a certain period of time, it's going to melt a certain amount of ice, turn it into water, and that's going to tell you how much energy was expended. Okay, so we can measure the energy an animal produces. Interestingly enough, a variant of this is still used in human metabolism. They still use this heat dissipation method. But it doesn't work for a lot of animals. So the next step was realizing that there is a particular ratio between the amount of heat produced by these animals and the amount of oxygen consumed. And it was the same as the amount of heat produced by a burning lump of coal and the amount of oxygen that was burned. And that answered the question about whether there was something unusual going on with living systems or if it could be simply viewed as a physical system, and it could. So therefore they said you can approximate how much energy an animal is using by how much oxygen it's using. The second thing I want to talk about, so that's the gas respirometry part. It's a standard thing we use now. Well, what is resting metabolic rate? Resting metabolic rate is the amount of energy an animal is using when they're not really doing anything. So they're resting, they're awake, but they're not active. They're not reproducing, they're not digesting their food, they're not trying to stay warm. So they're basic energy use. Resting metabolic rate is also known as basal metabolic rate. There's some differences we don't need to be concerned about. It's a measurement of how much energy you're using. And it's assumed that, so it's a good, the first point is it's a good comparative measure. You have a standard set of physiological conditions you can compare animals. The second point, it's assumed that resting metabolic rate has some strong relationship to total energy expenditure. That's why if you look at your smartphone and you have some sort of health app, you'll see a way of calculating your basal metabolic rate. So that is how much energy you should be expending if you're not doing anything. You put in your weight and your age, and that'll predict basal metabolic rate. The third point is because it's a standard measure, it's been used in the past. And this is the third important point. And this is Max Kleiber's mouse to elephant curve, which was produced starting about the mid-1940s. And it shows the very strong allometric relationship between resting metabolic rate and body mass across a huge range of body sizes. So this is the mouse to elephant curve. And so in theory, if you could measure resting metabolic rate in marine mammals, it's a pretty simple comparison to see are they different than terrestrial animals. You just make the measurement, throw them on this curve and see if they fall on the curve or above the curve. Well, in theory, it's easy. A little more difficult in practice. First of all, marine mammals don't like to sit in chambers and have their resting metabolic rate measured. And secondly, on a more practical basis, any answer you come up with is going to be hotly debated because of the implications for what it means for their food consumption. So there's a long history in bioenergetic studies of marine mammals of doing these measurements and then debating them. Which ones are valid, which ones were done correctly, which ones weren't. I'm going to show you some data that I didn't collect this data, but I sort of sifted through it. The bottom dotted blue line is Kleider's line for terrestrial mammals. The upper dotted line is 2 times that energy expenditure. It's a log. That's what you have to know. And these are some data I plotted for whales and dolphins. And without doing a serious statistical relationship, you'd be tempted to say, yeah, it looks like the resting metabolic rate of these marine mammals is higher than terrestrial counterparts. Now, part of the difficulty besides the politics is marine mammals aren't a physiological, well, they're a physiological group or an ecological group. They're not related to each other. So there's a huge diversity. So if you stick something like a sea otter on this figure, it's going to be way above the line because it's basically an aquatic weasel. If you stick a dugong or a large whale on there, it's going to fall way below the line because you've basically got an aquatic cat. So people argue you're comparing apples and oranges. And one of the ways that you can maybe do a more valid comparison is comparing the appropriate types of marine and terrestrial mammals. And this is a figure that was published a number of years ago. Comparing marine and terrestrial carnivores to marine and terrestrial herbivores. And the arguments this author makes is when you compare apples to apples and oranges to oranges, really marine mammals are no different than terrestrial mammals. I'd like to say after 30 years, the debate has come to some sort of conclusion, but I think you probably guessed that people are still arguing about it. What it did do, though, is this interest in resting metabolic rate opened up the sort of realm of the bigger field of energetics in marine mammals. And so people started looking at basal metabolism, and they also said, well, we can also measure or estimate different components of an animal's energy budget and come up with some measure of total energy expenditure. We can do this through bioenergetic modeling. Bioenergetic modeling is just, you put the different components of the energy budget of an individual together, and at the end you have a total energy requirement. Okay? And you can get very precise, very detailed models that will estimate total energy expenditures or requirements. The problem is, detail and precision does not mean accuracy. And these early models suffered from a lot of questions. They were based on very little data. So extrapolated from some pretty minimal data sets, quite often from terrestrial mammals, ironically enough. A lot of the components were actually measured as multiples or estimated as multiples of resting metabolism. Now we know physiologically that's not true. The cost of locomotion is the cost of locomotion. The other problem, of course, is if you have variance or inaccuracies in your measures of resting metabolism, that's just going to multiply through your model. Probably the biggest concern I have is the validation or lack of validation of these models, which was essentially what I like to cynically call the looks okay validation technique, which most people in this room that work with models know that's not a great way to validate a model. It was correct within an order of magnitude. So the question became, is there somewhere else we can use to validate these total energy expenditure outputs? And there was. So at the same time, scientists in the field were measuring field metabolic rate of different marine mammals. Field metabolic rate is the same as total energy expenditure. It's just a different term because it's the energy expenditure of what the animal happens to be doing over that time period. It's not detailed, we're just looking at total energy expenditure while it's doing what a marine mammal does in the field. We had a number of measurements, but they also had some limitations. They could only be done with a certain number of species. They were limited in the seasons that they could be done, which is very important when you're talking about energy expenditures. That's not constant through the year. And there was a question even whether the main technique, which was doubly labeled water dilution, was even valid for measuring energy expenditure in this group of animals. And I'll get into that later in the talk, the problems with that technique. So, question, is there another source of data that we have available that will tell us what the fish intake is of groups of marine mammals? Detailed records, perhaps over a long longitudinal period. That was actually a question. Okay, what about our friends at the aquarium? We've been keeping animals under human care for more than 5 decades. And one thing, regardless of the politics, one thing you can see about aquariums is they like to keep very detailed records about what their animals eat, to some level. So we have these measurements of what different types of marine mammals are consuming in this environment at certain stages of their life and even during the year. There's some problems with this though. Anyone want to suggest a problem with extrapolating what these guys are eating in the wild?
Dead. What they're eating is dead.
Dead fish? Absolutely. So no cost of capture.
There's no energy to capture things, right?
Yeah, no cost of capture. Yep. And no limits on what we can capture.
Probably.
Any other suggestions?
Daily activity is probably way different.
Okay, which would suggest what?
Lower, I guess.
Lower food intake. Yeah. And those are all valid concerns, absolutely valid concerns. Ironically, on the other side of the ledger is a conception that people massively overfeed marine mammals in aquariums. So there's questions about how valid it is, and we don't even know which way the bias might be. What's interesting is if we actually look at how across captive and wild terrestrial and aquatic mammals and do a large-scale allometric relationship, you get something that looks a little bit like this. So this is total food intake requirements. Here's Clymer's line. The black lines are for terrestrial animals, either captive or wild. There's one weird line at the bottom. And the rest of them, pretty well, yes, it's a lot of scale, so there's a lot of variation, but they sort of fall in to about the same area. Okay, let's put that aside. Let's assume that somewhere in here is a correct allometric relationship that is going to predict the food intake of an adult marine mammal based on its body mass. One of the problems we're going to face is the term adult. Right? Food intake is going to change depending on life history stage. How's that going to change? How does the food intake of a young animal compare to an older animal?
Goes to growth, the older animals will go to fat.
Right. So it's kind of one of these things that you have to get your head about, because actually food intake is going to increase as you get older, but your food intake per kilogram is going to decrease, primarily because of the cost of growth is being dissipated. And you'd think that we would, again, looking back at animals that have been held in aquariums for 50 years, we'd have tons of published data on how food intake changes with age. I mean, we're following these animals throughout their lifetime. Well, when I did a review, I was surprised to find that there's really only a handful of studies that have actually done this, and they show, this is body mass on the x-axis, daily food consumption as a percent of body mass on the y-axis, and they show the pattern we'd expect, but it's not necessarily a consistent pattern, and it's hard to sort of integrate between different species. But we know it's something we have to take into account. The other thing I want to point out is, when I talk about aquariums keeping very detailed records, unfortunately, sometimes the way they report it is kilograms fish, which we'll see becomes an issue. So they're not differentiating, they're just talking about this one ate 5 kilograms and it weighed 200 kilograms at the time. Because that's sort of how they used to think originally. The other problem is not only does energy requirements change with age, the energy requirements also change with time of year, right? Marine mammals, like most large mammals, are seasonal in nature. So how much food they need and how much food they're consuming changes during the year. An example of this is some of the original studies looking at resting metabolic rate noticed this at first, and they've been borne out through a number of species, that there's very periodic but predictable changes in resting metabolic rate during the year. These are usually tied to key life history events, could be the molt. This is some data I collected from ice seals, and the gray bar is their annual molt. And you'll see these oscillations in most pinniped species. These changes are not necessarily related to food intake. so they don't follow the same pattern. And what's more concerning is we're not even sure there's a clear relationship between changes in our resting metabolic rate, which we measure in the lab, and changes in total energy expenditure, which is one of our original reasons for measuring resting metabolic rate the first time, is we assumed the two were linked. We're also getting changes in some of the avert signals of that energy budget, food intake, and body mass. What was really interesting, one of the first things that discovered in the lab where I was doing my graduate work, is that when you feed a marine mammal easily, so it doesn't have to work for its food, you're not reliant on natural predictability or availability in the wild, it is still going to alter its food intake on a seasonal basis that is related probably to its opportunity to forage in the wild. They also have strong signals in body mass. Now, they kind of match up. You'd think when you're eating the most, you gain the most weight. When you're eating less, you lose weight. They don't exactly match up because marine mammals have this blubber reserve that kind of cushions changes in energy. These cycles are apparent in almost every species we've seen. They tend to become stronger as the animal gets more mature, although that's not always the case. Sometimes it's the reverse. We know it's tied to underlying hormonal changes, which are cued by things like the environment. So it's something we have to take into account when we're trying to figure out what is the requirement of that animal at that particular time. In the meantime, as we do all these types of studies, what we're doing is collecting more data on different components of the energy budget. So we have that original resting metabolism. We now have detailed studies on growth and reproduction and activity, whether we're talking about captivity or we're talking about wild. And what that's allowed us to do is progress from the point of building bioenergetic models to perhaps building realistic bioenergetic models, which is, I think everyone would agree, is a huge step forward. Now I talk about measuring all these things in captivity and in the wild. And one of the things we can do is come up with new ways of actually measuring energy expenditure in the wild. So these are just some illustrations of some ways that we can measure. We have accelerometry, just like you have on your smartwatch. Video recording, we can record when they're actually eating food and how much food they're eating and what size it is. We can use respiration rate to try to estimate energy expenditure, or heart rate. I love this figure because it's, I don't know if you can see, there's actually a little kid inside this model.
Yes.
That's what blue whale stuff, for scale. But I want to talk a bit about doubly labeled water, which I mentioned before. So doubly labeled water is sort of the, I won't say gold standard, the most normal technique used for figuring out total energy expenditure in vertebrates, whether we're talking about marine mammals or whether we're talking about humans. It's based on, without getting into the chemistry, on the differential turnover of two labeled components that you inject into the animal, and you can estimate then how much carbon dioxide they're producing. Sounds simple. There, of course, there's a lot of assumptions behind how that animal is behaving at that time. It's predominantly developed and used in human energy studies, so all those diet studies you read about in the press. And after decades and decades of that, they've gotten to the point where they have a single protocol they use that's going to give you an accuracy of 2% and a precision of somewhere between 2 and 10%. So you can have reasonable confidence in the numbers these come out to. But that's after decades of working with the same protocol on the same animal. It's not so simple for other animals, particularly marine mammals. And I had a graduate student who finished a number of years ago called Alex Dullman, who looked at this with northern bird seals. And he opened my eyes to the first big concern. When you read these papers about field metabolic rate determined by doubly labeled waters, they'll say, we did this, and we came up with this number. This was the energy expenditure. So we did the doubly labeled water thing. We're very careful on the chemical side, the injecting, the sampling, blah, blah, blah. We got good confidence in our chemical turnover. And then he went to turn that into energy, and he was amazed, and I thought he was wrong, and then he had to prove to me, that because there's a number of models that you can use and different sets of assumptions for how that animal is behaving, to get from the chemical to the energy, you have 36 different potential estimates. And these are not minute changes. Which model you pick can have huge implications on your final answer. They never talk about that in papers. In fact, usually they don't even explicitly state more than a few models that they put in to those calculations. The other thing which may or may not have been more concerning is when we actually tested the relationship between doubly labeled water and oxygen consumption in our fur seals at the aquarium. This is a really bad sketch of our experimental setup. We found on average, yeah, if you average it out over a whole year, you got within 2 or 5%. But the variance around that estimate for actual measurements during the year, because of changes that were occurring, were tremendous. It was the order of 30–35%, which when you go back and look at you look at the relationships in the literature, that's what you're finding. A pretty good overall average over some huge expanse, but the variation in that estimate is tremendous. Now, I also talked about measuring things on wild animals. Whether it's total energy expenditure or food intake or some sort of physiological parameter. And this is a common thing that we do with wild animals. We stick these biologging tags on the outside of them. We can measure activity, foraging success, diving patterns, physiological parameters. We can sample blood in real time. And in fact, it's so common on marine mammals now, one of the big things they're being used for is for oceanographic studies. So we're no longer worried about what the seal is doing, but we're sampling the oceanographic environment as it passes through. Here's the problem. We're measuring, let's say, energy expenditure on an animal that's very hydrodynamic, and we're sticking an external tag on it. And people have been reluctant to ask what that's going to do to our estimates. Well, in our lab and a couple of other labs, we're starting to ask that question. And it turns out not to be a big surprise if anyone who's watched Olympic swimmers with their skin tight, we don't want even a zipper showing. You stick a tag on a marine mammal, you're breaking up its hydrodynamic flow. That's going to have its effect on its behavior and also its total energy expenditure. It has to swim hard. I'm not saying don't use tags, but you have to account for that when you're looking at your calculations of what data you're collecting. So, let's assume that we got a handle on our individual energy needs for a moment, right? We know how much it's eating, we might know how much it needs at different times of the year. We can do different age groups, different genders, that's fine. Now we need to know what it's getting per fish. And of course there are many different, as they say on the dating sites, there's loads of different types of fish in the sea, or lots of fish in the sea. Okay, plenty of fish in the sea, that's it. And they're not all the same. And the problem is we can't really tell what an animal is getting out of a fish until we measure everything it loses. And so we have to measure how much energy it's losing so we can figure out what its net value is to the animal. And it looks great on a cartoon, very simple to do. In reality, it's a whole bunch of lab work, a whole bunch of literally getting your hands dirty as you're collecting samples. And it's a very tremendously tedious process, but doesn't make a difference. Okay, so here's an illustration. On the x-axis we have the energy density of the fish. Okay, on the y-axis we have the amount of fish, the kilograms required to fulfill a certain energy requirement. In this case, I picked 12,000 kilojoules per day. That's probably what a harbor seal needs. So if we look at something with a fairly high energy density, let's say herring at almost 8 kilojoules per gram, that's the gross energy, that's the chemical energy in the fish. We do all this lab work, how much of a difference do we make? Probably in the order of 12 to 15%. It seems like a lot of effort to make that sort of correction. Depends on what the question is that you're asking, what accuracy is. But if we go to the other end of the scale, we look at something, let's say like capelin, which has a high protein, low fat content. It's probably just approaching 3 kilojoules per gram. That's the gross energy, so you need a seal would need to consume over 4 kilograms. If we work it out how much it needs to eat based on the net energy, you can see there's a substantial change. That is going to become important when you're modeling. And not surprising, similar to what you see in terrestrial animals, there's a very good relationship between the energy density, actually it's the protein content of the fish, and how much you're losing. So the cost of the take home is the cost of digestion is really exaggerated as the quality of the diet starts to go down. The other thing we have to think about, even if we can do all these calculations and figure out what an animal needs over a year, we have these weird cases, like with many large mammals, where they're not eating consistently during the year. So we can figure out what their annual intake requirements are, But that intake isn't spread evenly over the year. So something like a male stellar sea lion here on the left, he's going to be fasting for 3 months of the year, defending his breeding territory, losing body weight. But he has to come out still at equilibrium at the end of some cycle, some annual cycle. So he has to make up for that over 9 months. Some whales are even more extreme. They might essentially fast, we're not sure if they completely fast, for 8 or 9 months, which means they have to have their entire annual food intake over 3 months. Now they can obviously do that, but it means the food intake is not even over the course of the year. And that means when you're trying to figure out the ecological impact of those intakes, that's going to depend on where they are in space and time. The other weird thing I want to point out with getting into too much detail is there's actually also an underlying relationship between how much food you need, or the prey quality, the prey patch quality, and how much energy you spend to catch that. And without getting into more details, it basically becomes tougher to make a living. You have to work harder. So as you're trying to fulfill those energy requirements on lower quality prey patches, you actually have to work proportionally higher to acquire that. That's going to increase your energy requirements. Oh, that's just what I said. Okay, so to summarize this sort of flurry of information, these are the take-home messages. So the accuracy of our point estimates of food requirements or energy requirements are very much a function of the method. And it's important to keep that in mind when you're interpreting the results of a particular study. They usually have, as a sort of grand stroke, their precision might be, their accuracy might be good, their mean accuracy, but their precision is usually pretty vague. Not everyone is a non-reproductive adult, so there's a whole thing about having to take reproductive history into account and age, right? That's going to vastly change your results. And underlying that, there's huge natural seasonal variation. This is not error. This is stuff you have to take into account that's going to make a difference on when you're calculating the impact. November in this area versus March in that area. And finally, I'd like to finish off the lessons with a total deviation from the main topic, and that's some suggested readings if I pique your interest on different aspects of this. And these are usually not intense scientific readings, but some things I would suggest you might want to go to. One is British author Steve Jones. I just finished a book a couple of months ago called No Need for Geniuses. And it's a very interesting read on the French scientific versus political revolution at the end of the 1700s. So, as many of you might know, I mentioned Lavoisier, who was sort of the shining star of French science, and of course he ended up on the wrong side of the guillotine. And the story that's always told in science history classes is the crowd shouted, but he's a genius, and somebody else shouted that the revolution has no need for geniuses. So that's a very good read. Another one that came out of, actually the Fishery Center is available online. One of Daniel Pauly's former PhD students, Kristen Kashner, had a very good, it's short, it's about 4 pages, discussion about the idea of competition between marine mammals and fisheries. It's probably about 11 years old now. It hasn't changed at all. And finally, something else that came up, if you really don't have an attention span for that, I would recommend a short animation presented by Jim Toomey. If anyone knows the cartoon Sherman's Lagoon, it's a marine mammal, a marine shark cartoon, and the Pew Foundation. And there's a YouTube, very good YouTube education. It's about 1:48 if you can spare the time, explaining what is ecosystem-based fishery management. So, if anyone has any questions or points they'd like to bring up for discussion, I'd be happy to entertain them with the class. Everyone just burnt out.
No, so
Yeah.
that was really interesting, and I guess you hinted at it over and over. But so, how wide is the variance in your estimates of how much marine mammals are eating? But at every point, you know what I mean? Like, does it just get
Yeah, does it get crazy? It certainly becomes unreasonable if you're talking about an annual estimate. So when I hear, I had some newspaper quotes about how much fish this population is taking. I would bet this is how much they're taking per year. And alarm bells in my head would be going off saying, okay, how are you coming up with that number? Are you taking a point estimate and just multiplying it? Because we know that's wrong. So there's a lot of natural variation that if you just sort of try to incorporate that, you should have huge error bars, right? Or your mean almost becomes meaningless. Do you know what I mean? Like you could say, okay, I've accounted for everything, and now I'll divide it by 365, or multiply it by 365 or whatever, and say, here's what they're eating a year. But is that a relevant number, even if it's a precise number?
Yeah.
If they're not eating it at the same place evenly over the year, then how can you talk about what the ecological impact is?
Because I know they're trying to incorporate in some places, like California, actually maybe in Canada as well, the biological acceptable catch or whatever, where they leave a portion of whatever it is, sardines or whatever, for sea lions. So, I mean, is that super uncertain? Like, how do they?
I would guess depending on what model they're using. So, the problem with most competition, particularly like on our west coast or with any fishing, is that marine mammals are as smart as we are as knowing when it's best to hit prey, right? So they're hitting, a salmon run for the same reason we're hitting a salmon run. They're aggregating. We're counting on that aggregation, they're counting on that aggregation. So they probably do need to eat a lot at that point. They're probably counting on that. So in some ways, our estimate, if we talk about an annual average, that might be a way of an underestimate at that case. We don't know. It depends how those estimates are put together. Some of these estimates, quite frankly, they're still going, well, this is the size, this is how they fit on fiber line. We think the average field metabolic rate is 4 to 6 times fiber. That's it. That's the level of estimation. Your average should be dead on. But is it a meaningful answer? Okay. Well, thanks for coming. Don't forget the midterm next week.